A couple of other substantial bioartworks is Eduardo Katz's the Alba, which is a really funny story. He was involved in a project with some scientists in France, and he met a group of rabbits that were being used as research subjects, and they had GFP, which is a gene from jellyfish, added into them through genetic modification processes in a sense so that they would phosphoresce. And this is used as a regular marker in scientific experiments to prove that a genetic modification has been done. But he agreed with the scientists that he would take one of these bunnies home and he would adopt it. He would name it Alba, and it would live out its life with his family. And that research animal would also become a pet, sort of transforming the relationship between us and those animals. And of course, when he came back to pick up the rabbit, the boss of the research institute said, "No way, Jose." And part of that is due to bioethics and health and safety, but part of it is also because they knew it would be very controversial. So the real artwork in the end became Eduardo Katz running around the world with these posters saying "Free Alba," and he created international outrage and student protests, et cetera, et cetera. But the ironic thing is that he has photoshopped Alba to a bright green color, and that's not what GFP looks like at all. It sort of phosphoresces, and you'll see I have some GFP work later in here. It phosphoresces in the fluorescent light, and it's only the skin and the eyeballs that would do that, and it's not the fur. We already talked a little bit about tissue culture and art projects, and I'll touch on them later. And the last thing I do want to talk about is Critical Art Ensemble, a very famous American group of bioartists who do a lot of work that challenge sort of corporate relations with the biotech field. Here in Free Range Grain, they did a project where they built a reverse protocol that would allow them to determine whether or not food contained genetically modified ingredients. And they traveled all over Europe testing food products that had been stamped as approved as not containing GMOs, and found that over 85% of those products did in fact contain some genetically modified corn as part of their product. It's important to note that this field, although all of us are incredibly responsible and follow the rules, there is a lot of concern about non-specialists engaging in the sciences, and a lot of misunderstanding. So Steve Kurtz, one of the members of Critical Art Ensemble, had some very serious legal troubles in 2003. He is a professor at Buffalo, and unfortunately his wife died. She was also a member of Critical Art Ensemble. Her name is Hope Kurtz. And when he woke up in the morning and found her having had a heart attack and died next to him in bed, he called the paramedics, and the paramedics came into his house, and they saw petri dishes growing bacteria, and then they also saw a lot of books on bioterrorism, because he was writing a book on bioterrorism. And he spent the next 4 years of his life in court. He was charged originally with bioterrorism. That was eventually reduced to mail fraud, because some of the bacteria had been sent through the mail from a scientist that he was collaborating with. In the end, the charges were dropped. They were not found to be guilty in any way. But I want for you guys to really understand that when I talk about the sort of political nature of these extreme interdisciplinary actions, that this is like an extreme example of the type of outcome that can come from non-specialists working at high-end biotech projects. So that's my introduction, and now I'll tell you a little bit about the work that I do. The work that I'm most well known for is a project called Biotechnica. It's a collaboration between myself and Sean Bailey from 2000 to 2007. And we created a fictitious bioengineering corporation that produced organisms based on consumer demand. Our product was, we did a lot of research on sort of corporate representations of the biotech sector. And we found that the representations of the research were often very tame and cute and polite in comparison to the research that they were doing in terms of their scientific journal papers, etc. So we decided that we wanted to bring some of the meatiness and unruliness and bloodiness of biotech research into a more public light, and we made a product line of teratomas. And the teratomas are a real object of biotechnological study. They are known also as an endodermal sinus tumor or a dermatoid cyst. And these tumors are essentially filled with germ cells that have a chromosome break that prevents them from growing in a rational way. So essentially, this is it's a germ cell tumor, very similar to a fetus, and the cells differentiate as they divide within the host organism, that they differentiate in an irrational and grotesque manner. They do have a nervous system, sometimes hair, nails, teeth, sometimes sensory responses after they've been removed from the human body. So scientists are interested in the teratoma as an instance of reverse cellular differentiation within the human body, a really important key to life, a really important key to growing organs for people who might need an organ transplant. But also the Christian fundamentalist right is really interested in the teratoma as an ethical source of stem cells. And they would argue that these cells could be used for research and that teratomas do not intrinsically have a soul in the same way as the fetus might. So it's with that sort of like really charged component that we decided that Biotechnica would, in a very fake way, at no point did we sell teratomas, propose to sell teratomas basically as consumer organisms. And I have to tell you, it was shockingly successful. We thought that it was really going to leverage a great response from our viewers, whereas we had a number of people who actually wanted to invest in the company and thought it was a really appropriate strategy. So we actually had to push the project further and further as a means of upsetting and creating a more critical relationship between the general public and our fictitious corporation. So on the left here you can see that we had an online version where you could generate your own teratomas based on the ingredients that you inserted. At the top we did a series of public autopsies where we made teratoma sculptures utilizing meat products that we had bought from the butcher. And then in 2003 we met Oran Katz and Willet from Tissue Culture and Art Project at the University of Western Australia, and they challenged us to try it for real. And this is a really key point in our project, but also a key point in my artistic career. Up until that point, I'd had a very third-order representational relationship with the biotech sector. And their argument was that our critique was really rooted in, sort of filmic, popular culture representations of biotechnology, and that although we could read the scientific journals, until we had tried some of these techniques for ourselves, we could not have the same level of sort of complex relationship with these processes. And I have to tell you, it was a very difficult choice, and I was a vegetarian when I went there. But they were right. And so in 2004 and in 2006, we traveled to Australia, where we trained hands-on in tissue culture and tissue engineering technology, which these are the images you can see here. And what we did is we grew our own mouse teratoma cells, and these are teratoma cells from a mouse embryo. And it's funny because they don't look like that, but they're actually the exact same thing. All of these cells are differentiating into different types of cells. And they're really unruly and grotesque in the way they clump up on top of one another, and they sort of starve out each other and die off on a fairly regular basis. More healthy, normal cells, like muscle cells, for example, will lay in beautiful rows and have almost a mechanistic appearance to them, whereas the teratoma is irrational and grotesque even at the microscopic scale. So with Oran and Willet, we created a project called Teratological Prototypes, where we made, as a sort of, again, a representation of Biotechnica product line, teratoma sculptures. Okay? This project was funded through SSHRC, Social Science Humanities Research Council of Canada, and was subject to human research ethics approvals until, because originally we were going to use our own cells to grow in the sculptures. We thought that was more ethical, that we could consent to donating our cells. But in the end, we had to go with animal-based cells just for health and safety reasons. So this project resulted in 2006. We showed it at ICEA in San Jose, California, and at a number of other galleries worldwide, where we created a portable laboratory environment that was a sterile environment. And within this space, we had a functional tissue culture and tissue engineering laboratory. And within that space was the Biotechnica teratoma sculptures called Teratological Prototypes. And it's right there. And it's a bioabsorbable polymer, which is sort of like a fuzz, that we had cast into the shape of a teratoma form. And the teratoma shape was derived from having scans of our bodies, and then we put them into the computer and we morphed them into sort of this grotesque, irrational shape. And then we outsourced that and